having the opportunity to talk a little bit about these unique stop signs or checkpoints that a cell could participate before it commits to the next phase, right? We said that cells, like you and me, have different events in their course of their life, and there are points in the cycle where you can hit the brakes and abort the mission, okay? And these checkpoints are strategic. There are three well-documented checkpoints. The first one is located in the midpoint of G1. The second one is located at the end of G2. And the third one is located inside the M phase or mitosis. And these different red blocks that you see here represents an opportunity for the cell to stop whatever it's doing, completely give up, and then go through a process of cell death and disintegration. And so that the cells, if there are any errors, it doesn't keep on building up, right? So that's why when you think about cancer, what that really comes down to is that when a cell has missed the opportunity to abort any mutation and it keeps on dividing, right? So what causes that to happen? A lot of the research is pointing at, well, maybe during these important checkpoints, important roadblocks, there are proteins that play a role to govern what gets to go and what doesn't get to go. And in cancer cells, then those cells that should have those little proteins that regulate them are not there or not working, right? So that's what causes an issue. And there are numbers of factors out there, but one of the more accepted way to see this is that over the course of your life, your cells and your DNA are constantly bombarded and exposed to damage, right? So the more you live, inevitably, the more damage your cells are going to receive. That means you're going out to the sun, right? You're eating more kinds of foods, you're putting more chemicals in your body. Over the course of your life, the longer you live, the more chances you're exposing your body to damage. Every time you get damaged, your cells need to be able to rectify and fix those damages, right? So let's just say you work in a coal mine and you've been in this very toxic environment for 10 years. So you're accelerating your body's ability to be exposed to damage and then healing. And the more you heal, probably the more chances you're going to find an opportunity for error or mutation to occur, right? And that's really the thought process that I want you to have. And so if a cell is not ready to divide, right, what happens is you have this opportunity to stop it. You can actually block it off right at G1. You could block it off right at G2. Or even during mitosis when it's splitting, you can also still abort it. But then if it skips all three of these, then you're pretty much not going to be able to stop the problem. There's one word here on this slide that I want to highlight called apoptosis. And this is a word you might not hear all the time. But apoptosis refers to programmed and dedicated cell death. So when a cell in our body has discovered a mutation or some kind of a problem and is trying to very controlled and targeted destroy that single cell or that set of cells, that's called apoptosis. So that's actually a good thing. This is a very good system that your cells can do and isolate. And it makes sense because let's just say you're doing your rough draft and you're writing, and as you're writing, right, you realize, oh my God, I don't really like this topic, and I've spent maybe 20 minutes, 30 minutes of outlining what I want to write, right? So you can either try to massage what you have written into another topic and fit something in, or just start from scratch. And that's essentially the same concept. Your cells can either repair the damage of the mutation, or it can just straight up wipe it out and restart the whole thing. So chromosomes is what contains all the genetic information, and this is the DNA inside the nucleus. So what happens here is that DNA is coiled, it's tightly packed into the structure called chromosomes, and you'll see some of this in the lab when we do the isolation experiment, but this is what the chromosomes look like. You see the famous X and Y chromosome, and you hear about that because the X and Y chromosome is when the chromosomes are condensed very tightly into this almost X and Y shape. All right, and when you look closely at a chromosome, you will see this kind of fuzzy texture. It almost looks kind of fuzzy, and the reason why it's fuzzy is because there are tons and tons of little strands of DNA that are just wrapped around together like a yarn. Okay, it's just packed together. And it's interesting here because these chromosomes are organized densely packed together so that when they divide, you can easily pull them apart. When they're not dividing, the chromosome is going to be more loosely scattered and almost kind of like a bowl of noodles kind of setup. All right, so it's a little different depending on when that cell is either in division or if the cell is not undergoing division. Altogether, humans, you and me, we all have a total of 23 pairs of chromosomes in every single one of our cells, except for our sex cells. So the female egg cell and the male sperm cell will only have half the number. All right, and one half will then get matched up with the other half when the sperm meets the egg. These concepts are pretty, there's a lot of content here, but this is well documented, well studied. And one of the most fascinating parts about cancer and DNA is that on the DNA, on the chromosomes, they have discovered that there are these sections of nucleotides or bases that are repeating at the end of each chromosome. And these are what they call telomeres. There's a lot of research on this, very fascinating stuff. But telomeres basically indicate how often the cells go through division. So they begin to shorten every time a cell cycle of division occurs. And when they get too short for replication, the cell begins to cell death. And that's why we talked about the apoptosis, where the cells are marked for the programmed self-destruction. So in other words, you can look at a cell and analyze the telomeres just to get a feel for how old or how much damage the cell has gone through. So let me give you another application to the story. So why is it important to avoid all the carcinogens, avoid the damage, avoid mutation? Well, because when you are exposed to viruses, when you are exposed to radiation, when you are exposed to toxins and carcinogens, you are giving your body a higher opportunity to make a mistake. And when your body makes a mistake, there's probably going to be a chance where some cells are going to die and some cells might become mutated and become cancerous. That's really the bottom line. So when these chromosomes begin to divide, they don't just randomly split, right? There has to be a process of controlling them, putting them together, and they form this pair called the homologous pairs. Is important because when these pairs come together and they split, the mom and the dad, right, the sex cell, the sperm and the egg, they each have a set of chromosomes. You can't just have two, and right, you can't have a sperm that has more than 23 sets, and then you can't have an egg that has more. And altogether, when they combine for you and me, we have total 46, right? So you can't have a cell that has 50. And when you have a cell that has more than the number of pairs or less than the number of pairs, then we have issues. We have what they call trisomy, Klinefelter's, Down syndrome. But that's when you have these issues that become genetically predisposed, and then the outcome is that the offspring will have oftentimes some delayed development issues, mental capacity development issues, and of course even deformities. So that's when it gets really scary. And the way they know this is they did some really, really heinous experiments, and most of this research was done back in World War II. So I want to just kind of give some recognition to those times that, you know, there was a very dark time in history, and during the German reign and the Nazi camps, there were some very, I wouldn't say really breakthrough experiments, but they did some very notorious research on how chromosomes and how DNA and how the way the cells split. They exposed tons of pregnant females to different kinds of chemicals. And when they exposed the females to chemicals, obviously those chemicals went through into the uterus. They went into the developing embryo of the baby. And so when they exposed them to like thalidomide, these chemicals, the infants that were delivered basically were missing limbs, and you can look this up as well. It's very scary. It's very depressing. But they did experiments where they exposed females to chemicals, and then the offsprings would not have an arm or not have a leg. And that's just what happens, is that when you temper with the chromosomes, you will then change the outcome of the phenotype or the expression of what the body becomes. Very scary stuff. And these are all real experiments that were done. And obviously those were considered, wartime and very inhumane and unethical in terms of the codes, but that was what the some of the Nazi experiments involved. And let's backtrack a little bit. So now that the chromosomes are paired up in the homologous pairs, and that means they have two sets of the same exact type coming together. They might have different coding, but you have one set from your dad, one set from your mom, right? So let me just draw this out. Okay, this might be a little bit easier. So let's say you have a sperm cell. Okay, this is a sperm cell, and this is an egg cell. Right, so just to kind of give you a feel for this is the female. Right? The female is the symbol right here, and the male is on the right. And now imagine these two cells come together. Right? You finally find the partner of your dream, and the sex cells come together, and they are able to then fertilize and form an embryo. Right, so the sperm eventually is going to tack onto the egg cell, and they fuse together. And then they begin dividing. All right. So in other words, they divide. So what's important to remember is that the sperm contains half the number of the total cells in a normal cell. So you have 23, and then you have 23 in the egg. So when they combine together, the embryo is going to have a total of 46. All right. So every single cell in our body has 46, except for your sex cells, and your sex cells go through a slightly different division to get that. It goes through reduction division. And as the cells are dividing, what happens is the chromosomes line up and they pair up together. Okay, so that's called the homologous chromosomes. And then as they begin splitting, those homologous chromosomes are then going to form what they call the chromatids, and they get pulled apart. So there are three, I would say probably three versions of DNA that gets confusing. Okay, so as you're reading the text, or if you're reading the content, or if you're doing the smart reading, the homework assignments, the three words that tend to throw a lot of students off, and I'm going to write this out. The first one is called chromatin. All right, chromatin. The second one is called chromosome. And the third one is called chromatid. All right, so it's like, oh my God, this sounds like the same thing, and I'm gonna have the urge to wanna think of them as the same stuff, right? So chromosome, chromatin, and chromatid. Right? And you think, what the heck is the difference? Right? Obviously, spelling is different, but let's start with the first one. So chromatin, the one on the upper right that I just erased by accident, chromatin is when you have DNA that is in the loose and uncondensed format. It's scattered all over the place. It's like spaghetti. It's like noodles. It's loose. Chromosome, the one that you hear more about, the one on the top of this PowerPoint, is going to be when DNA is condensed down and you have a shape. You have the X or Y shape. And then the final version, chromatid, TID, is when the cells and the DNA are getting ready to divide. They become pulled apart as single strands. All three of those words, chromatin, chromosome, and chromatid, are referring to DNA only at different phases. And that's the trick. All right? Otherwise, as you're reading, or if you don't have a good background in biology and you see the words, you will get a little confused, and it might throw you off a little bit. All right, so let's get started with mitosis. So mitosis is the famous phase of our cells when they begin to divide. And I'm going to use this illustration to show you the different phases. And during lab time, you guys will get a chance to draw it out. You guys will have some beads to mimic the movement of the chromosomes. So hopefully that will make it stick a little bit better. But the story begins with interphase, where the cells are getting ready, right? Getting ready to amp up for this big phase of its life. And during interphase, these eukaryotic cells tend to begin to get thicker, get bigger, organelles duplicate, and oftentimes you won't really see the DNA, and this is when you call the DNA chromatin. All right, chromatin. So this version that you see is like, oh, it's kind of all over the place. It's just scattered all over the place. All DNA is inside the nucleus. Nothing really shape or visible shows up. Chromatin, right? Chromatin. And then once you begin to condense down into prophase, which is the first phase of mitosis, then you start to see the chromosome version, right? You see the lines. You see the little strands coming together. And then the centromere gluing them together. Right? So that's going to be the difference. Chromatin versus chromosome. And you'll see that the hallmark events, if you will, for prophase is a few things. Number one, you will notice that the chromosomes begin condensing. That's number one. You will also notice that these little proteins called the spindolic fibers begin to generate, and then the centrosomes are very, very prominent. These are little proteins that will guide division. These are really interesting to talk about. There's a lot of research about how they work, but they have to be precise because the last thing you want is to split your DNA and have an uneven amount of DNA being pulled apart. Right? That's very scary to think about. Think about just pulling DNA, right? You're actually literally dividing your cells into two, but let's say you mess up, right? How hard is it to make, or if you ever bake cookies and you try to make every cookie the same size, and without the right tools and you're just using your hands, it's pretty hard, right? You might have some that are smaller than others, some that are bigger than others, some have more chocolate chip, whatever it happens, right? So it could be error prone. So over here in prophase, you'll see that the cells begin to condense down and then DNA is more visible. The centrosomes begin to move apart and the spindolic fibers are going to be growing. And then the last, I would say, hallmark phase to this, which you don't really see that well in this picture, is that the nuclear envelope begins to disintegrate. So this membrane outside of the nucleus, I'm gonna highlight this with the pink color. Right here, you see that there's a little bit more fragmented cracks along the membrane. And this is important because of the fact that, well, at this point, you're going to get ready to allow the DNA to move and bind, right? So you want the DNA to split. And so now prophase is really this very important first step that gets the tone ready, gets everything ready so that the DNA and the cells could do its job during the split. So you'll see right here that as it transitions to the end, or what they call late prophase, the complete nuclear envelope is gone, right? The DNA is literally just exposed into the cell. And then the spindolic fibers have fully adhered along the plane of the chromosome. And then at the two ends and two poles, you have these centrosomes very clearly on opposite ends of the cell. All right, so those are probably going to be what you want to look for, right, when you look at a cell. So if you have a chance, when the chance comes in lab and you look at the microscope and you're looking for those cells that are dividing in different phases, look for those characteristics that I just said. Okay, and that marks the end of prophase. Now, prophase is going to transition into my metaphase. Let me zoom up a little bit. Metaphase, so P, M, A, T. All right, so that's your acronym, PMAT, for mitosis. Prophase, metaphase, anaphase, and telophase. So metaphase is going to be when the chromosomes are all fully aligned inside the cell's equator. Very, very noticeable, pretty easy to spot. You can see the chromosomes just all in one straight line. Nothing too crazy, but they're all along the equator plane, and then now they're getting ready to split. Right? So this one is a pretty easy one to spot. The third phase, anaphase, is going to be now a few things happen. So you can see now, instead of it being called a full chromosome, these single strands are what we call chromatids. Right? They're being pulled apart. And you can see this. You can see closely that when they line up, you want each side to have the same exact split, right? Because you don't want one side to have more than one chromosome, right? That's really the whole point. You want to have an even split. You want to have a fair trade. And these spindle fibers are going to attach to the chromosome, allowing it to disassemble to each pole. And ideally, in a healthy, happy outcome, is that cell 1 is going to have a set of chromosomes, cell 2 has the same set. And earlier when I was saying when these homologous chromosomes line up and they don't pull the right way, or if one cell gets one more chromosome, then you get those genetic disorders like what I said, trisomy 21, right? Or Klinefelter's, or double X syndrome. Like there's a lot of conditions where you have additional chromosomes, and whenever you have more chromosomes in a cell, it's always bad. Right? Don't ever think like, oh, that's going to be how X-Men or mutants are created and you get superpowers. Not really. Most of the time when you get extra chromosomes, you get the bad case scenario. You get something that doesn't work, or a deficiency, mental retardation, deformities, that kind of stuff. And then the final phase of the story, after it splits into two and the migration begins, is then hopefully you have a successful divide into telophase. And telophase is the final phase of mitosis where you have a split. And now there's also another phenomenon that occurs, which is the pinching off of the cytoplasm. And as you can see right here, as I'm drawing these lines in the middle, the cells are going to begin to invaginate in, twist apart, and allow the cells, the parent cells, to begin to turn apart from each other. And the cytoplasm begins to pull apart. And this is kind of a point of no return. So going back to what we were talking about with the checkpoints and cancer, what's important is that there is a checkpoint at that metaphase, anaphase position, but the moment it goes past anaphase, after this goes on, it is already the point of no return. So it's like you're committing to something, right? So once you get through that pulling apart, the cell doesn't just get a chance to go back, say, hey, let's hit the brakes and let's rejoin again and then go backwards. It doesn't do that. All right, and that's why cell division is a process that's highly engineered, very, very precise, but also very delicate. All right, so let's go ahead and do a question today. So at which of the following checkpoints is a cell's DNA checked for damage, and if found, the cell is sent back to G0 phase? All right, so use what you just heard and try to make the best choice and best guess possible. This is the new question for the day, so make sure to answer this. At which of the following checkpoints is a cell's DNA checked for damaged? And if there is damage, the cell gets sent to G0 phase. You can use your notes, you can read your text, you could use Siri, you could use Google, you could discuss. All right, so the whole point is to attempt the question and try to rationale what you think is the answer. Whether it be right or wrong, it doesn't matter. You are very welcomed, and the whole point is to make the mistakes now so that when you see a question similar to this on the exam, you get that right. That's the goal. All right. All right, so we got 1 more minute to answer the question, folks. 1 more minute. Let's get the ball rolling. And I only see 50 answers. We have close to 80 people in the room. Another 45 seconds or so. If you need more time, you can just post in chat or raise your hand. All right, I can add more time to this. It's all good. All right, last but not least, let's go ahead and get your answers in. All right, stop answering. And let's take a look at the responses here. All right, the responses is 51% said G2, 27% said G1, 20% said M phase, and then 3% said none of the above. All right, so the answer to this question is G1. All right, so if you go back, remember, let me just show you where the answer is. If you go back a little bit, right, to the chart. Right, so I'm just going to retract back right here, right? So G1, right? So we are, if a cell stops over here and you're going back to G0, the phase is in G1, right? So just look at the pictures. When you're studying biology and when you're using the text, one of the reasons what I will probably say in defense for some of these textbooks, I know some books, some classes utilize the lower cost or zero cost books, but I believe that some of the publisher texts that do come with a price, they provide some really solid images and good graphics. And you probably also know that creating images and creating designs and drawing pictures out, they require skill sets, right? Sometimes it requires some coloring, sometimes it requires some design. And that's why I really recommend you, when you're reading the homework assignments and doing the pre-works, make sure to look at the pictures, all right? So the pictures are what you're really paying for. The text isn't that important. You can type it out, you can find that information, but you don't see these images just randomly out on Google, because these are all copyrighted. These are from the source that we're using, okay? So just a little tip there is to utilize the images as you're studying and as you're reading. All right. Okay, so we had that question in. Let's go to the next question. All right, we did this one already. Last question for the day. So go ahead and answer this question. The replication of genetic material occurs following a successful passage of which checkpoint? So when does replication occur? And after which phase? M, G1, G2, or G0. So go ahead and look at the picture, right? So I just told you how to find the answer. Look at the picture and find when does replication occur and what's the phase before that. Look at the picture. The answer is there. All right, so you're part of the training that you need to learn is to be able to dig around, find the answer, right? Go through the pictures, go through the paper, go through the articles, and find the answer. All right, another 35 seconds. The time is clicking. The clock is ticking. All right, 10 seconds left, so go ahead and answer your question. Give it a best attempt here. All right, so let's take a look and see what the responses are for this one, right? So the responses that you guys all decided to post here is we have 19% of you say M phase, 10% said G1, and a whopping 58% said G2, and then 12% said G0. All right, so what is the answer? Let's take a look. Well, the answer is G1. All right, so we got a little bit confused here, so let's go ahead and backtrack and take a look at the chart again. So we'll go back to the image. Right, and when does replication occur? Well, replication occurs in, where does it occur? The S phase, right? The S phase. So what phase has to happen before the S phase? G1, right? So what phase has to be successful? It has to do its job. The cells have to grow, double in size, organelles have to enlarge before you can go through replication. The answer is G1. All right, so the step right before it. G2 is a step right after it, right? So this is the chart to remember. It's G1, S, G2, M. G1, S, G2, M. Right? So that's the way you should draw up the chart. And again, I'm going to refer everybody to make advantage and take use of those key images when you see them. When you read the pre-work, when you're watching along in Top Hat, you need to focus on the images. Right? So that also means this image that we just went over with the mitosis, there will be plenty of questions that can come from here that could be on our next exam. Right? So just key questions, and you can answer all of them from the images. All right. Okay, so I think that's a pretty good stopping point there for today. We have another 2 more sessions on the cancer, so we'll keep the ball rolling next Tuesday. But I want to pause here, open this up for questions. If you have any questions, please let me know on what we just went over. Any questions about the metaphase, anaphase, prophase, telophase, anything about chromosomes or chromatin, anything about cell growth, any general questions or just concerns at all from the group? You can post it in chat or just any other questions. And then I did say that I will be giving the extra credit on Tuesday, right, for the webcam. Today it looks not as convincing. We have 82 people. I see like 1, 2, 3, 4, 5, 6, 7. I see like 7, 8, 9 cams. So today is a little weaker on that effort. But don't worry, there will be other opportunities for you to get some extra credit. So stay tuned. Clarying asks, what did the chromatin mean again, and what phase can we see it in? A good question. So let me go ahead and answer that question. So go ahead and I think your question was in private to me, but there's a question that was posed to me here, and I'll just post your question, Clara, in the group chat. It's all good. What did chromatin mean, and again, what phase can we see it at, right? So just kind of backtrack here, right? So when you're looking for these answers, the first thing you should do before you dive into reading a lot of stuff, and I'm sure you don't like to read too much content, is to look at the pictures, all right? So over here, if you look at the phase of mitosis, and you zoom in, the word chromatin is right here. Chromatin is when you have DNA that is found in the loose and scattered sparse form, right? It's literally all over the place inside the nucleus. And chromatin is just DNA. That's all it is. It's just a loose version of DNA. Whereas chromosome is the condensed version of DNA, right? It looks like the X and the Y's. Okay? And then chromatin is only present in interphase and all the other phases, not during mitosis. All right? So every time your cell is not in mitosis, your chromosome is pretty much, your DNA is pretty much in chromatin form. Think of chromatin, chromosome, and chromatid like different layers or different tiers of Super Saiyan, right? You have Super Saiyan 1, Super Saiyan 2, Super Saiyan 3. It's just different forms of the same thing.